Hello everyone. In today's lecture, we'll be seeing about the brief discovery of enzymes, the chemical structure, concept of transition state, activation energy, classification and nomenclature of enzymes. Enzymology is the study of enzymes. Enzymes are biocatalysts which speed up the rate of metabolic reactions in our body. In human cells, there are about 40,000 different enzymes, and the enzymes help the chemical reactions or the physiological reactions happening in the body to take place well at the normal body temperature. The enzyme diastase is the first enzyme to be discovered. French chemist Anselm Payen in the year 1833 discovered the enzyme diastase. Louis Pasteur made an immense contribution with fermentation, and he named the force or the factors concerned with the fermentation as ferments. Wilhelm Kuhne, 1877, German physiologist, he used the term enzyme. In the year 1897, Edward Buchner of the University of Berlin, he found that the sugar was fermented even when there were no live yeast cells in the mixture which is used. He won the Nobel Prize for the discovery of zymase, the enzyme which brought about the fermentation of sucrose. Chemically, enzymes are proteins. There's an exception which is ribozyme. Being proteins, enzymes are thermolabile, they are sensitive to pH, they are colloidal in nature, they can be denatured and precipitated with salts, solvents, and other reagents. Some examples of enzymes, even in the glycolysis, we have hexokinase, phosphoglucoisomerase, or phosphohexose isomerase, several enzymes. In the process of digestion, we also have pepsin, trypsin, etc. Coming to the concept of activation energy, in these graphs you can see that as the reaction progresses, the conversion of substrate or the reactants to products take place through an higher energy level called as the transition state. Though the product is placed at a lower energy level than the reactants, the reaction doesn't proceed downwards. The reaction proceeds upwards first, and then it comes down and forms the product. So the higher energy level which is needed is called as the activation energy. In an uncatalyzed reaction, in a reaction where there is no enzyme involved, higher amount of activation energy is needed. Whereas, if enzymes are involved in the reaction, the amount of activation energy required is lower. At the activation energy, the molecules, the substrate molecules are in the transition state. It is an unstable, short-lived chemical structural state of the substrate. We can again see here with an example, the reactants being carbon dioxide and water. The red line in the graph shows the reaction without enzyme. The reaction proceeds without enzyme, but you see, you require a higher level of activation energy. But when an enzyme is involved in the reaction, you require a lower amount of activation energy. Okay, so the enzymes act by lowering the free energy of the transition state. There could be several reasons for that. The enzyme might be putting a stress on the bond which is facilitating the breakage of bonds in the reactants. The enzyme might bring two reactants to close proximity in space and maintain proper structural orientation for the reaction to occur. Or the enzyme might also give the good microenvironment by donating or accepting protons in the reaction. So the conversion of substrate to product is illustrated here with a simple flowchart. It's not directly like enzyme to substrate forming enzyme to product. We have intermediate states. There is an enzyme substrate complex. The enzyme substrate complex is highly unstable and it is in the transition state. and then it gets converted into enzyme product complex. Then the product dissociates from the enzyme. So enzyme is not used in any of the reaction, it is just a catalyst. Nomenclature of the enzymes and the classification. Trivial names initially used for the enzymes like pepsin, trypsin, etc. They do not give any information about the source of the enzyme or the type of reaction it may catalyze, and the trivial names may also vary. So, schematic name has been proposed and it is used. According to the International Union of Biochemistry and Molecular Biology, the name of the enzyme will have two parts. The first part will indicate the name of the substrate, and the second part will indicate the type of reaction. And it ends with the suffix ase. For example, lactate, substrate name lactose, dehydrogenase, reaction is dehydrogenation, ase ending for denoting the enzyme. This is a systematic nomenclature. This nomenclature and the classification of the enzymes was determined by the Enzyme Commission in 1961. Okay, so in the case of this classification of enzymes, there are there are six classes, sorry, there are six classes of enzymes which are used. The classes are named 1 to 6 in that order. Number 1 are the oxidoreductases, number 2 are the transferases, 3 hydrolases, 4 lyases, 5 isomerases, and 6 ligases. So the Enzyme Commission has designed an EC number or Enzyme Commission number for all the enzymes. The number is 4 digits separated by a dot or a period. The first digit represents the main class, which will be 1 to 6, which we just saw. The second digit indicates the subclass. The third and the fourth digits are denoting the subclass and the further subdivision or the serial number. Generally, the B and C digits describe the reaction, while the last digit is used to distinguish between the different enzymes of the same function based on the actual substrate in the reaction. So there are generally six classes of enzymes, and the nomenclature involves four parts. The enzyme nomenclature as a number. Now, the six classes we just saw, what type of reactions do they catalyze? Oxidoreductases, this is the general scheme of the reaction. It could be dehydrogenation, oxidation, okay, reduction. Transferases are involved in the transfer of groups. Hydrolases are involved in hydrolysis reaction. Lyases are the enzymes which are used for synthesis. And isomerases are those which bring about changes in the structural part of the substrates, and ligases are enzymes which are used for joining the bonds or forming molecules. These are the six classes of the enzymes in a brief nutshell, the type of reaction, and an example which is given there. Okay, thank you very much.